Hey everyone, my name is Yen-Kai. I did a master's on space biology at the University of Auckland. Today, I'll be talking about some aspects of space biotechnology, a little bit on what was done in the past, what's happening now, and how it could potentially advance. And so today's talk is about the different areas of space biotechnology and how it benefits people down here on Earth. Even though it sounds niche, space biotechnology, like general biotechnology, is a very broad term. When we talk about space biotechnology, this can include hardware, software, and materials that are used to grow and analyze biological processes. And these can include bioreactors, the use of machine learning and sensors. There are also biological molecules that can function as indicators or be used for environmental changes. I separated biosatellites into its own thing because it's something that requires a combination of all the different aspects. There are also ground-based modeling instruments for simulating space conditions, but I won't be talking about that today. Ever since the biosatellites in the late 1960s and Skylab, biological experiments have been carried out. This hasn't stopped during the Space Shuttle program and is even more true today on the International Space Station and on CubeSats. So why bother spend money on space technologies? Technologies are just, well, technologies. Just because something was designed for space doesn't mean it can only be used for its specific use in space. I won't be talking about all the examples, but some examples include the memory foam, which was originally designed to keep pilots cushioned during flights. And so now it's used in beds, couches, and other everyday items. The sky blue LED was developed to help plants grow in space, but now it's also used to keep humans awake at work and at home. NASA aims originally designed ways to protect astronaut helmet visors from scratches, but now it is also in consumer sunglasses and for ski goggles. The CMOS image sensor are smoke cameras made for interplanetary missions, but now it's also in your cell phones and also used in medical imaging. So it can also be used to benefit humanity for healthcare, in healthcare. Formulate was initially designed for recycling agent in long-term space travel, but it's also very beneficial for infants and general dietary supplements. The ethylene scrubbers that was originally made to help plants grow without filters in space stations is also now used to remove particles, pathogens from the air, so air purifiers. Fluorescent paint were initially made to assess for genetic damages from radiation. And so it was used to, so it's also used for DNA abnormalities in cancer. It's used in agriculture and in industrial safety. The food sent to space are often checked to make sure that it's completely pathogen free. The procedures are now used as a standard for food safety testing in countries all over the world. For the biosatellite section, I won't be talking about the specific technologies involved, but I will talk about some of the learnings and what they were used for. So the biosatellite program in the late 1960s, they all relied on sample retrieval for analysis. Unfortunately, the Biosat-1, the vehicle propulsion malfunctioned. So it ended up slowly decaying until it disintegrated in the atmosphere. Biosat-2 had to have an early return due to a tropical storm threat. But otherwise, it was a successful experiment that contained plants, frogs, insects, and several microbes. Biosat-3 was returned early because the monkey that was on Biosat-3, the monkey's health had deteriorated, and it died one day after recovery. So while this mission had failed its scientific agenda, it did point out to the importance of substantial testing before flights. And then after more than 4 decades later, we have GeneSat-1. And so GeneSat-1 was launched in 2006. It measured cellular growth and metabolic activity of E. coli. And so it showed that there was a slower rate of growth in spaceflight, and this is likely because of convection and sedimentation in microgravity, means that nutrient and waste exchange only occurred through diffusion. So what GeneSat demonstrated was that autonomous biological research in spaceflight is indeed possible. So PharmaSat took on the lessons of GeneSat and tested how antifungal agents affected the yeast Saccharomyces cerevisiae's growth. So while in spaceflight, the yeast did have a slower rate of growth, but after the data normalization, they showed that there was no difference between how the antifungal agent affected the ground control and spaceflight samples. But otherwise, this was the first free-flying pharmaceutical dose response system. The OREOS was the only CubeSat mission with an astrobiology research focus. At 650 kilometers Earth orbit, it experiences 15 times the radiation than that of the International Space Station. This saw bacillus spores revived not just after 14 and 97 days, but also after 6 months in orbit. And it also exposed several compounds to full spectrum solar illumination. And so only the aromatic hydrocarbon registers changes. So this showed that the bacillus spores and also the organics that some biological organisms and molecules were in fact quite stable and in space. And so SPORESAT looked at the changes in calcium channels on a aquatic firm. They did this to prove that gravity affected cytoplasmic calcium concentration. So this turned out to be true. But otherwise, in the process, they did make these mini centrifuges, which generated artificial gravity in orbit. The ECMSAT was, I guess, heavily dependent on PharmaSat on design and on spare hardware. But instead of testing on a fungus, we're looking at testing on antibiotics, the gentamicin on E. coli. And so it showed us E. coli, still also had same as GeneSat, E. coli had a slower metabolism and microgravity. But what it did not show, or contrary to expectation, microgravity did not enhance the uropathogenic E. coli's antibiotic resistance. In fact, they were more susceptible. So this was quite surprising. Otherwise, Artemis I, which is expected to be launched sometime next year, hopefully. It will take Biosatellite, the CubeSat is the Biosatellite beyond low Earth orbit and around the moon. And so over there, it will be exposed to a lot more radiation than in low Earth orbit. Also, not on a separate satellite, Artemis I will also be carrying experiments on fungi, algae, and plants, and this will be inside the Orion capsule. But summarizing the BIOS CubeSat, in general, there were severe limitations in the type of analysis that can be carried out. They all use similar sensors, and a lot of them only looked at how growth is affected in spaceflight. But on the International Space Station, there are instruments which allow for more complex analysis. There are a number of technologies for DNA sequencing, and so these could be useful for looking at how radiation affects an organism, or what kind of organisms are growing in space stations. There's also the E-nose, which is good for detecting volatile molecules. But some of the most in-depth analysis are omics analysis, so transcriptomics, proteomics, metabolomics, because these yield us the most important information. And so these still rely on sample returns. Also not true of the, I do need to point out the qPCR can do targeted transcriptomics. But there are upcoming capabilities such as the GEM, the gene expression measurement module. And so this does look at a broader range of gene expression transcriptomics than qPCR. There's the MicroTitan, which looks at automating the extraction process. And so there is a trend looking to remove the need to do sample returns. In terms of commercial success stories, something I found is that it is that someone on Earth has to benefit from the technology. And so this could include fixing a medical issue, or for scientists to do research in space. So you won't really see a business succeed on creating food for space exploration, but you can potentially succeed if you make a business case on how it can be used by people on Earth. An example being the baby formula. Another example could be the lab-grown meat, but you don't have to have a space perspective on that. But I think some of this can change with space tourism. And so once space tourism becomes a regular thing, I think there could be a case to be made for food production in space for the tourists. And another capability I saw could be interesting to see in the future is that you could have capabilities that, or technologies that includes biological processes. And so in A, you have the traditional control system that you can see in systems engineering. And on the right, you have the biology in the loop. So where you can have a electromechanical external input, which will result in a feedback from an internal biological sensor. So bottom left, C is just an example of a normal biological process. But on the right, the D, it is where you have a mix of biological and abiotic components. And so both on the right, B and D, it can potentially be used to facilitate unnatural biochemical production, or to compensate for certain biochemical abnormalities and diseases. Also, as to summarize, Space Biotechnologies has spin-offs with the technology itself have an Earth application. These past 2 decades have seen limited progress in biosatellites, and this is mostly due to the type analysis that can be conducted. Also, if there are good upcoming systems in the ISS or will be seen on the ISS as well. That's all from me. Thank you. I hope you enjoyed that.